Hello and welcome to Sam online class. Friends, today we shall discuss about beta-pleated sheet. Beta-pleated sheet is a protein secondary structure that was identified by Pauling and Coil. The name beta has been given because it is the second protein structure that was elucidated by them. Beta-pleated sheet has a zigzag appearance. The formation of beta-pleated sheet depends on intermolecular hydrogen bonding, although intramolecular hydrogen bonds are also present. Beta-pleated sheet is formed whenever there is a parallel alignment of polypeptide chain in a plane. Beta-pleated sheet is of two different types. One is parallel beta-pleated sheet and the other is anti-parallel beta-pleated sheet. In both parallel beta-pleated sheet and in anti-parallel beta-pleated sheet, these are the participating polypeptide chains. These polypeptide chains have been aligned parallelly so that hydrogen bond will be formed between the NH group and the CO group of adjacent polypeptide chain. What is the difference between parallel beta-pleated sheet and anti-parallel beta-pleated sheet? In parallel beta-pleated sheet, the C terminus of the participating polypeptide chains are on the same edge, whereas the N terminus are on the opposite edge. That means in parallel beta-pleated sheet, the polypeptide chains are arranged in the same N to C terminus direction. But in case of anti-parallel beta-pleated sheet, the C terminus of these participating polypeptide chains are not on the same edge. But in anti-parallel beta-pleated sheet, the C terminus of the alternate polypeptide chains are on the same edge. That means in anti-parallel beta-pleated sheet, the participating polypeptide chains are arranged in opposite N to C terminus direction. This is the difference between parallel beta-pleated sheet and anti-parallel beta-pleated sheet. In both parallel beta-pleated sheet and anti-parallel beta-pleated sheet, the R group of the constituent amino acids project alternately above or below the plane of the ring. This is a polypeptide chain. Here, this R group is projected above the plane of the ring, but the next R group is present below the plane of the ring. So, in parallel beta-pleated sheet and in anti-parallel beta-pleated sheet, the R group of the constituent amino acids are projected alternately above or below the plane of the ring. There are some amino acid residues which favor the formation of beta-pleated sheet. They are glycine and alanine. There are a number of differences between alpha helix and beta-pleated sheet. First difference is in beta-pleated sheet, the polypeptide chains have extended conformation, but in alpha helix, the polypeptide chain is tightly coiled. Next is the axial distance. The axial distance between adjacent amino acids in beta-pleated sheet is about 3.5 angstrom, but in alpha helix, the axial distance is 1.5 angstrom. The beta-pleated sheet is stabilized by intermolecular hydrogen bonding, but the alpha helix is stabilized by intramolecular hydrogen bondings. These are the differences between beta-pleated sheet and alpha helix. Silk fibroin is an example of protein that contain anti-parallel beta-pleated sheet. Now we shall discuss about some other protein structure. One is random coil. Next we shall discuss about gamma helix. Last one is beta tone or beta band. What is random coil? When the polypeptide contain adjacent bulky residues, for example isoleucine, or charged amino acid residue like aspartic acid or glutamic acid, the force of repulsion between them cause the polypeptide chain to assume the configuration of a random coil. This is the structure of a random coil. So this random coil configuration is formed whenever there are adjacent bulky amino acids like isoleucine in a polypeptide, or when there are adjacent charged amino acid residue, for example aspartic acid or glutamic acid. Another protein structure is gamma helix. Gamma helix is a highly hydrogen bonded structure which is insufficiently stable to be used in protein structure. Another protein structure is beta torn. Beta torn is also known as beta band or hairpin band. It is also known as reverse torn. Beta torn is found where a polypeptide abruptly reverses its direction. For example, this is a polypeptide chain. This polypeptide chain has abruptly reversed its direction. So where the polypeptide chain abruptly reverses direction, at that place we can see the presence of beta torn. Beta torn usually connects the ends of two adjacent segments of anti-parallel beta pleated sheet. This beta torn, which is also known as reverse torn, is usually present on the surface of protein. Glycine and proline are the two amino acid residues that are often found in the beta torn. Glycine is preferable in the beta torn because it is small, and proline is also preferable in the formation of beta torn because the peptide bond involving the amino nitrogen of proline readily assume a cis configuration that is more amenable to formation of beta torn. And in the beta torn, the CO group of N amino acid residue is hydrogen bonded. In the beta torn, the CO group of N amino acid residue is hydrogen bonded with the NH group of N+3 residue. So beta torn is a protein secondary structure that is found where a polypeptide chain abruptly reverses direction. Most of the globular proteins are compact. This is due to the formation of beta torn. Because of this, the polypeptide chain abruptly reverses its direction that help in the formation of a compact structure. And this beta torn is a tight torn, approximately 180 degree. And the formation of beta torn involve 4 amino acid residues. So friends, this is about various protein structure, that is beta pleated sheet, random coil, gamma helix and beta torn. In the next video we shall discuss about some other protein structure. So please watch my next video, and for more updates please subscribe my YouTube channel Sam Online Class. Thank you very much.